So let's talk about photosynthesis. Well, plants use photosynthesis to generate carbohydrates like glucose. And here we can see a couple of photoautotrophs that can carry out photosynthesis. Cyanobacteria, for example, produces a lot of the oxygen we find in our atmosphere. And the energy stored in carbohydrate molecules that are produced from photosynthesis are able to pass through the food chain. So, what is photosynthesis? Well, it's a process that traps sunlight and by doing so is able to fixate carbon dioxide, which is behaving inorganically, into an organic molecule like glucose, a carbohydrate. So, it's this process, this energy conversion, where we store solar light and we convert it then into chemical energy. This occurs primarily in leaves and in the chloroplasts of plant cells we find in these leaves. There's plenty of adaptations for photosynthesis. And the majority of carbohydrate molecules, like glucose, are produced through photosynthesis. Organisms convert solar energy into chemical energy to build glucose or carbohydrate molecules. All organisms carrying out photosynthesis require sunlight, and energy holding these carbohydrate molecules together is actually released when food is broken down. We break down carbohydrate molecules to produce ATP. That's a process we've already looked at called cellular respiration. But the energy that is transferred from one organism to another is actually going to initiate itself or begin in photosynthesis because that's where the carbohydrate that's broken down to produce ATP comes from in photoautotrophs. So whether it's directly or indirectly, photosynthesis provides most energy required by living things. Photosynthesis also results in the release of oxygen into the atmosphere. Here we can see a simplification of the chemical equation for photosynthesis. We see that sunlight is required to convert carbon dioxide with the help of water into glucose or sugar and oxygen. Oxygen is actually considered a byproduct of photosynthesis. Let's look at some definitions. An autotroph produces its own food. Plants, certain types of bacteria and algae, they would be autotrophs. And they're primarily photoautotrophs, which are autotrophs, so organisms that produce their own food, that use sunlight and carbon from carbon dioxide to synthesize chemical energy in the form of carbohydrates. Now, we're heterotrophs. Humans and all other animals are heterotrophs. We're incapable of photosynthesis, and we obtain energy and carbon from our food. We break down carbohydrates, so we consume other organisms, whether they be animals, plants, or a combination. So we directly or indirectly depend on autotrophs, depending on our diet. So here's the balanced chemical equation for photosynthesis. 6 carbon dioxide molecules plus 6 water molecules yields, thanks to the energy of sunlight, 1 glucose molecule, which is C6H12O6, and oxygen, 6 oxygen actually. So this is the process of photosynthesis that can be represented by an equation. But you can see that it's really the conversion of sunlight or light energy into chemical energy. We're going to store some of this light energy in the bonds of glucose, or C6H12O6. And we actually convert carbon dioxide into sugar. We convert a carbon-containing molecule that behaves inorganically, carbon dioxide, into something that we consider highly organic, glucose, or C6H12O6. So let's look now at these structures in photosynthesis. And most photosynthesis takes place in the leaves of plants. And leaves have a couple of different structures. They have outer epidermal cells. They have inner mesophyll cells. They have in their lower epidermis these holes or these spaces that allow gas exchange, that allow carbon dioxide to enter the leaf and oxygen to leave. These spaces are called stomata. And in all autotrophic eukaryotes, photosynthesis takes place in the chloroplast, an organelle of a plant cell that contains chlorophyll. Chlorophyll is a group of pigments. We have chlorophyll A and B. They have ranges in pigment shade from kind of yellow to green. And we find them in algae, green plants, cyanobacteria. And they are necessary for photosynthesis because chlorophyll is the light-absorbing pigment in chloroplasts that make up the plant cells of leaves, the primary structure in a plant responsible for photosynthesis. So let's look closer at the structure of a leaf. We have the leaf itself coming off of a stem, If we look closer at the leaf, it has upper and lower epidermal cells. Inside, we have mesophyll cells. The upper cells near the upper epidermis that we still consider mesophyll cells are nice and tightly packed together. These mesophyll cells are called the palisade mesophyll cells, and they sit above the spongy mesophyll cells. We see in the lower epidermis that we have a stomata, or an opening, and that allows CO2 in and oxygen out. We can also see, if we look closer, an individual plant cell. These are plant cells that contain not only central vacuoles, but also chloroplasts. And chloroplasts have an outer and inner membrane. Within that membrane, there is a stroma. The stroma is a fluid-filled cavity that contains a lot of these discs. Each individual disc is called a thylakoid, but a stack of these discs are called a granum. So a granum is a stack of thylakoids. These grana sit within the stroma, or the inner cavity, of a chloroplast, and the chloroplast has an inner and outer membrane, and we can find chloroplasts in plant cells. Plant cells like the palisade mesophyll cells under the upper epidermis of a leaf. So those are just structures to look at, and these structures are where photosynthesis takes place. Again, primarily in the chloroplasts of plant cells under the upper epidermis. And that makes sense because most of the light would hit the upper epidermis, not the lower epidermis. So continuing on, both the upper and lower leaf surfaces consist of an epidermis. These tend to be transparent. So it really is the pigment chlorophyll that's giving leaves their color. Photosynthesis in plants takes place at the chlorophyll-containing organelles we're calling chloroplasts, and mostly they're contained within internal leaf cells, like the palisade mesophyll cells that sit under the upper epidermis. And they contain chemical reactions able to convert energy in sunlight into stored energy in sugars. So here we can see leaves, and just to simplify, these are the location of photosynthesis. But to really look at specifically what happens, we have to look at individual plant cells. Here's another view of internal leaf structure. So we have the inner mesophyll cells, the upper and lower epidermal cells. The lower epidermal cells contain stoma, that is the singular for stomata. These are openings that allow for gas exchange, allows for CO2 to go in and oxygen to go out. The cells around the stoma are called guard cells, and they can help open and close these gaps. We also have certain types of mesophyll cells, and it's the nicely packed palisade mesophyll cells where most photosynthesis takes place using the chloroplasts that we find in plant cells, because these chloroplasts contain chlorophyll, the pigment that absorbs light. So here is the mesophyll cells containing chloroplasts. So this is one individual palisade mesophyll cell containing many chloroplasts. And we could look closer at these chloroplasts and look at their structure. So on the outer surface of both epidermal layers of a leaf, we also have a cuticle. If you've ever seen certain types of plants, they might have kind of a shiny layer. That's actually a transparent, waxy, or waterproof covering called the cuticle, and it's supposed to prevent evaporation of water. And that allows leaves to obtain carbon dioxide for photosynthesis from air through their stomata without losing too much H2O when the stomata are open. So we have this kind of protection of evaporation of water because water is needed in the process of photosynthesis. So don't forget that leaves are the primary location for the cells that perform photosynthesis. Make sure you review their structure. Mesophyll cells are in the middle, epidermal cells are on the outside, and the waxy coating that prevents water evaporation is the cuticle. Here are pictures of stomata, and around the stomata are guard cells. So we can see open stomata on the left and closed stomata on the right. The mesophyll cells are generally either loosely or tightly packed. It's the tightly packed palisade mesophyll cells near the upper epidermis and the loosely packed lower spongy mesophyll cells near the lower epidermis. And these cells are responsible for photosynthesis, especially the upper ones near the upper epidermis. So they're loosely packed layers of internal leaf cells where chloroplasts are located. So photosynthesis can take place here. Other areas near the lower epidermis and the spongy mesophyll cells are locations for gas exchange. We also saw vascular bundles in the image of leaf internal structure. And those are just veins transporting water, minerals, sugars throughout the plant. Think of them as the plant's blood vessels. So there has to be some way to get nutrients from the roots up towards the stems and finally the leaves. And they're surrounded by protective supportive bundle sheath cells. So here we can see kind of the vein or artery of a plant. They generally contain things called a xylem and phloem, which are just tissue that we find in these bundle sheath areas for these vascular bundles. So chloroplasts are organelles with a double membrane enclosing a fluid called stroma. We've covered that basic idea. Well, let's look at the stroma. What is it like? Well, it's a gel-like fluid found inside the inner membrane of a chloroplast. Interesting that chloroplasts, like mitochondria, both have double membranes. They both have an outer and inner membrane with an inside area that contains a fluid. Here we have the inside area that contains a fluid, and we're going to call that fluid the stroma. And we find within this stroma thylakoids. And thylakoids are those discs, and the discs like to stack together. The stacks of discs or stacks of thylakoids are called a grana. And thylakoids are suspended in the stroma of the chloroplast and are the sites of the light reactions of photosynthesis. And we'll talk about what happens in photosynthesis. We'll look at the actual chemical reactions. One is going to be light-dependent reactions, oftentimes shortened and called the light reactions. And those light-dependent reactions have to occur in the thylakoids because that's where the chlorophyll is. Chlorophyll is embedded within these thylakoid membranes. So again, we're looking at leaf as a structure, but we're about to look at microscopic structures and how exactly one single organelle can contain structures that process this complicated metabolic pathway. So just keep all this in mind, but we're about to look even closer at individual cells. And not just the individual cells, but the chloroplast, one organelle that's within these plant cells. So we're going to look at this specialized structure called a chloroplast, an organelle found in plant cells. And we can find chloroplasts in plenty of plant cells, but especially the mesophyll cells within leaf epidermi. So here is an electron micrograph of a chloroplast. A little bit harder to see the outer and inner membrane, but we can see two layers. And the inner layer has these stacks, kind of stacked discs. Well, the stacks are called grana, and what the individual discs are thylakoids, and they're suspended in the gel-like stroma. So here's a drawing of a chloroplast, so we can see everything in a little bit more detail. So here we see a stack of discs, a grana, and each disc is a thylakoid, and it sits within the inner membrane in a fluid stroma, and they've also labeled the outer and inner membrane. So remember, in order for the reactions of photosynthesis to take place, we have to have carbon dioxide. We have to have water. We must also have light energy. Sunlight is needed to convert light energy into chemical energy. So we'll combine carbon dioxide, water, and sunlight to create glucose, and then have oxygen as a byproduct. So we're going to need light energy to be held somehow. And the way we do that is we trap this light energy using chlorophyll that's located in the thylakoid membranes of a chloroplast. So these are the two reactions or two parts of photosynthesis: the light-dependent reactions and the light-independent reactions. The light-independent reactions are called the Calvin cycle. I like to call it the Calvin cycle because it fixates carbon dioxide. And the fact that carbon dioxide and Calvin cycle both start with C helps me remember what it does. The light-dependent reactions occur in the thylakoid membrane, so the individual membranes of those thylakoids or those discs. That's because in the thylakoid membrane there is chlorophyll, so that's the location where light is absorbed. So chlorophyll absorbs energy from sunlight and that then gets converted into chemical energy with the use of water. There's some energy products produced from the light-dependent reactions, as well as some ATP that help power the Calvin cycle, or the next step. The light-dependent reactions also release oxygen from hydrolysis of water as a byproduct. So it's from the light-dependent reactions we get oxygen via the splitting of water. The Calvin cycle, or light-independent reactions, don't necessarily happen in the absence of light, they just don't need it. But it does take place outside of the thylakoid membranes. It takes place in the gel-like fluid we call the stroma. And the chemical energy from the light-dependent reactions is utilized here to drive the capture of carbon from carbon dioxide molecules. And we're going to see a process called carbon fixation occur. Carbon dioxide molecules will be converted into glucose. So we'll convert carbon dioxide into glucose, which is an organic molecule. So we're going to convert an inorganic molecule to an organic one. And that's the process of carbon fixation. So the carrier molecules that transport energy from one place to another that we've seen in cellular respiration, like NADPH and FADH2, they're present, and ATP was created in the light-dependent reactions. And that's going to be utilized to create even more energy to power the Calvin cycle. But remember, we're not focused on making energy. We just need to make energy to power the two parts of photosynthesis. The main product we're going to see from the Calvin cycle is glucose. So the photo refers to the light reactions. Synthesis refers to the Calvin cycle. So photosynthesis is a process that captures light energy using the light-dependent reactions to next synthesize glucose in the Calvin cycle. So here's what's going on. The light reactions are made possible by molecules or pigments that we know are called chlorophyll, anchored within the membranes of the thylakoids, those discs. And we're going to use the products of the light-dependent reactions to power the Calvin cycle, which occurs in the stroma instead of the thylakoid disc. So you can see the energy from the sunlight hits the thylakoid disc. It would be trapped by chlorophyll, and this process would also split water to produce oxygen. This process would produce some ATP and electron carriers as a result, and those things would go through the Calvin cycle and then be recycled back to the light-dependent reactions. The Calvin cycle, which doesn't need any sunlight, doesn't need that light energy, but can occur in a light or lit environment, will use CO2, run it through a couple of steps to produce a 3-carbon sugar. So you actually have to do the Calvin cycle a couple of times to get 2 3-carbon sugars that we will combine to make a 6-carbon sugar that we know as glucose, or C6H12O6. So how is light energy converted to chemical energy? We saw the processes that do it. We looked at where they happen in leaves, and then specifically in chloroplasts, and then where in the chloroplast? Well, the sun emits energy within a broad spectrum of electromagnetic radiation. And the electromagnetic spectrum ranges from really short wavelength gamma rays through to ultraviolet or longer, higher wavelength gamma rays. And these are ones that we consider visible all the way up to infrared light and the longest wavelength radio waves. So we have to think of light critically. You could get into quantum mechanics or get into things that are quite complicated, but let's just simplify everything. We can think of light as little packets of energy, and the packets are called photons. But if we were to look at an individual packet, we would see within this packet waves, because light travels as an electromagnetic wave. Invisible light has wavelengths with energy strong enough to alter the biological pigment molecule, like chlorophyll A. That's the key light-capturing pigment molecule in chloroplasts. It absorbs violet, blue, and red light, and then reflects green. The green light being reflected is why leaves appear green. And chloroplasts also contain other pigments. They're not all chlorophyll. Those are called the accessory pigments, and these absorb additional wavelengths and other light energies, and then they transfer them to chlorophyll A. These accessory pigments do not always emit or reflect yellow to green light. Now, chlorophyll B does absorb blue and red and even some orange wavelengths of light missed by chlorophyll A. But the most common accessory pigment is actually carotenoids, which kind of reminds me of carrots, which is helpful because it reflects yellow to orange light because it absorbs all blue and green light, so it appears yellow or orange to our eyes. So here is a graph showing the electromagnetic spectrum, and we can see the general wavelengths for chlorophyll A, chlorophyll B, and carotenoid. And in the autumn, there's actually more abundant green chlorophyll breaking down than the carotenoids breaking down, or the anthocyanins, which kind of have a purple color. So it's the carotenoids and anthocyanins, these accessory pigments, that are left over in kind of mid to late autumn. So it's the pigments that reflect yellow and orange wavelengths, or the ones that reflect red to purple wavelengths, that are left over in dying leaves, whereas the chlorophyll that reflects anywhere from yellow to green pigments breaks down quicker. So that's when we see leaves change color in the fall, because the leaves of the chlorophyll is degrading, and what lasts longer are the accessory pigments. So here are just some examples. We could see kind of yellow, purple to green, and these are all emitting or reflecting different wavelengths of light. So it's actually possible to separate the pigments used in photosynthesis via paper chromatography, which we gave a shot last week. And paper chromatography is a technique using a solvent to separate mixtures or compounds onto filter paper. Now, we didn't quite have these types of results, but you should have seen chlorophyll B and A separate out. Chlorophyll A has more of a blue color, whereas chlorophyll B can have anywhere from a yellow to green color. Xanthophylls and carotene should have orange to red colors. All right, so let's move on and look at the specific reactions in more detail. The light reactions that occur in the thylakoid membranes happen with photosystems, and photosystems are a group of molecules, a cluster of chlorophyll and other molecules that are inside the thylakoid membrane. So they include the chlorophyll that absorbs the light, but also these other integral proteins. And we actually discovered photosystem 1 before photosystem 2, and then we found out they go in the opposite direction when we're looking at these light reactions. And each photosystem, whether it's 1 or 2, has a unique electron transport chain associated with it, or located adjacent to it. So in the thylakoid membrane, we have photosystems that contain chlorophyll, and that's where light is absorbed. Then that high energy is passed as electrons to associated electron chains that are related to photosystem 2 and 1. And remember that an electron transport chain is something we saw with cellular respiration. It's a series of electron-carrying molecules within a membrane that transfer electrons and release energy at each transfer. So this is a series of electron carrier molecules, and they have reaction centers that will help transfer this high energy from sunlight. There's also a pair of specialized chlorophyll A molecules and a primary electron acceptor molecule embedded in this complex of proteins. So within the thylakoid membrane, the overall path of electrons is as follows. Remember, they discovered photosystem 1 before photosystem 2, but we found out later that the process of photosynthesis, especially the light reactions, is in reverse. So we actually see that light hits photosystem 2 before going to electron transport chain 2, and then being passed to photosystem 1, and then going to electron transport chain 1. And then NADP plus is generated, which will eventually be recycled back into NADPH. So the light energy that is absorbed by a chlorophyll molecule and is passed along a pathway to other chlorophyll molecules does so in the thylakoid membrane using photosystems and electron transport chains. And that energy culminates in a molecule of chlorophyll that gets excited enough to push these electrons into the reaction center. And that energy excites one of its electrons enough to leave the molecule and be transferred to a primary electron acceptor. And that's one of the processes responsible for splitting a water molecule. And that splitting of the water molecule will need to be replaced once one is donated. And the oxygen and hydrogen ions are also formed from splitting of water. And again, we're going to see the formation or generation of these free hydrogen ions that have to be pumped somewhere and build up a high concentration. So let's look at what's happening here. We have photosystem 2 being hit by light. That light is exciting electrons that move into a reaction center. So from photosystem 2, the electrons move from a reaction center into electron transport chain 2. They'll eventually move into photosystem 1, where they're hit by even more light and excited again into electron transport chain 1. During this process, we see the splitting of water to generate oxygen plus some free hydrogen ions. These hydrogen ions can be transferred into the space known as the thylakoid space, or the inside of the thylakoid membranes. And then at the very end of electron transport chain 1, NADP+, one of the electron carriers, will combine with the free hydrogen to be regenerated into NADPH, which can then be used again in the Calvin cycle. We also see that ATP synthase is still present. ATP synthase is at the end of both photosystems and both electron transport chains, and it's going to use this buildup of hydrogen ions in the thylakoid membrane space to power the production of ATP by combining ADP and inorganic phosphate. So this is the same thing we saw with cellular respiration, except now we have photosystems to accept the light because they contain chlorophyll. We get the transfer of electrons, the buildup of hydrogen ions, and then the transport of those hydrogen ions through ATP synthase to create ATP. So the ATP and NADPH produced by the light reactions occurs here at the thylakoid membrane. And then we'll transfer the NADPH and ATP to the light-independent reactions to power the Calvin cycle. So the energy of electron movement through the thylakoid membrane created an H gradient, and that H plus gradient, or proton gradient, is going to be used in the process of chemiosmosis, and we're going to move that gradient through ATP synthase to generate ATP. And it's the generation of ATP that produces the ATP that powers Calvin cycle, and there's also NADP+ that goes to the Calvin cycle. So where all of this is occurring, the two photosystems in green with their electrons and reaction centers, and the two blue electron transport chains, is in this membrane of the thylakoid. It's occurring all over these membranes of the thylakoid. And the thylakoid are the discs that stack together to form a granum. And the H+, the protons that are being pumped to the thylakoid space, are being pumped right here into the middle. And they'll be pumped out into the stroma through ATP synthase to produce energy to power the Calvin cycle. So the light-dependent reactions harness energy from the sun, produce ATP and NADPH in order to power the Calvin cycle. So we're going to move to the Calvin cycle, and that occurs in the stroma. So here is the Calvin cycle. It's essentially a process of carbon fixation. We're going to turn carbon dioxide molecules into sugar. For every one round of the Calvin cycle, we create one 3-carbon molecule, and we need 2 of those and combine them to make glucose. So the Calvin cycle essentially has 3 stages. We use the enzyme Rubisco to incorporate carbon dioxide into an organic molecule. That's the carbon fixation. Rubisco is a really interesting enzyme because it actually takes inorganic carbon dioxide and adds it to other things in order to make it something we consider organic. It's a very quick process, and so it's a very efficient enzyme to fixate carbon so quickly. Step 2, the organic molecule we've created from carbon fixation has to be reduced. And that process requires even more ATP. That reduction will break down NADPH we got from the light-dependent reactions into NADP+ and free protons. Then we have to regenerate RUBP, a molecule that is going to be needed to generate or create Rubisco. Rubisco is actually going to combine carbon dioxide with RUBP to form carbon fixation. So without RUBP, Rubisco cannot act. We must regenerate it at the end or after one round of the Calvin cycle. So the Calvin cycle essentially captures carbon dioxide. It uses the ATP and NADPH from the light reactions to power this process, and it's accomplished in the stroma. The stroma is where the Calvin cycle occurs, and it's a series of reactions. The enzyme Rubisco can actually carbon fixate. Carbon from CO2 is incorporated or fixed into a larger organic molecule. We essentially use Rubisco to combine carbon dioxide and RUBP. And then RUBP and the combined carbon dioxide will break down or be reduced into PGA. And PGA essentially can then leave the Calvin cycle, but that's a 3-carbon molecule. So we would need 2 of those to create glucose. And because of the generation of this 3-carbon PGA molecule, the Calvin cycle is sometimes referred to as a C3 pathway, a carbon 3 or 3-carbon pathway. The PGA molecule could leave the Calvin cycle, but it's actually easier if we convert PGA into G3P, because we can just put 2 G3P together to create a glucose molecule. So it's really the conversion of PGA to G3P that produces the product that leaves the Calvin cycle. So G3P is the product that leaves the Calvin cycle. 2 rounds of the Calvin cycle produces 2 G3P. We combine those together to make glucose. And then the end of the Calvin cycle regenerates RUBP, which will eventually be reunited with more carbon dioxide to generate a carbon fixated molecule, thanks to Rubisco. So it's the remaining G3P molecules that exit the cycle. Some of the G3P actually has to be used to regenerate RUBP. So let's look at that. Our light reactions that occur in the thylakoid membranes split water and produce ATP and NADPH. They power the Calvin cycle, which fixates carbon dioxide by connecting it to RUBP using Rubisco. That process creates PGA molecules, which get converted into G3P molecules. G3P molecules are 3-carbon sugars. So the Calvin cycle spits out at least 2 3-carbon sugars if we do it twice. And we can combine those 2 3-carbon sugars into glucose to create C6H12O6, or the carbohydrate we were after all along. So that leaves us on to some other plants. There's other plants that live in harsh conditions, like those in the desert. This cactus actually had to evolve variations and reactions outside the Calvin cycle, and those variations are in order to increase efficiency and help conserve water and energy. So there's some other pathways besides the C3 pathway. There's the C4 pathway, the CAM pathway, which stands for the crassulacean acid metabolism pathway. And these are essentially pathways that are adapted by some species of flowering plants that need these alternate pathways in order to circumvent wasteful photorespiration, or the loss of certain gases and or water. So when plant stomata are closed in hot environments to prevent water loss, oxygen happens to build up in the plant cells and RUBP combines with it, which is not great because we'd like it to combine with carbon dioxide, because carbon dioxide is what we need to be fixated, not oxygen. So that's wasteful. That's a process called photorespiration. So if we can't get rid of oxygen and water, we kind of have this unintended reaction of photorespiration. So this process prevents the Calvin cycle from synthesizing sugar because RUBP combined with oxygen instead of CO2, and plants could die under these circumstances, so they needed to evolve alternative pathways. So we have, therefore, the C4 and CAM pathway. In the C4 pathway, the Calvin cycle puts Rubisco in its own separate cell. And Rubisco, therefore, is not exposed to any accumulated oxygen. So you're going to have, for example, a mesophyll cell and then a kind of separate bundlephyll cell. And it's where the bundle sheath cell is located that the Calvin cycle takes place. And then oxygen builds up in the mesophyll cells. For the CAM, or crassulacean acid metabolism pathway, CO2 is harvested at night but stored to be fixed during the day. So when the stomata are closed and oxygen is accumulating, even though it's at the same place, the process of the Calvin cycle won't happen right there or at that time. So we have kind of a same place, different time scenario. So even though oxygen is accumulating, it's not during that time that it would react with RUBP because the Calvin cycle is not trying to happen. So this prevents the accumulation of oxygen from getting in the way of the Calvin cycle, these modifications that plants have taken on, like crabgrass and corn, daisies, pineapples, succulents, and cacti. So what do plants do with the glucose once they make it? What do plants do once we've done the light-dependent and light-independent reactions, once the Calvin cycle has produced its glucose? Well, it's broken down during cellular respiration. Plant cells essentially create their own glucose, then use it in cellular respiration to produce their own energy. We also can put a lot of extra glucose together and link it in a plant to form starch, which is a plant storage molecule for glucose. They can link it together to form cellulose, which is a major component of plant cell walls. So they can form both nutrient storage or structural components of their own cells. And these components may be used in amino acids, nucleic acids, nectar, fruit. So certain parts of photosynthesis can be broken down or utilized in other parts of the cell. So in the carbon cycle, which we've looked at before, we saw that the reactions of photosynthesis and cellular respiration were responsible for cycling carbon dioxide as a carbon-containing component throughout the environment. And so these two processes share reciprocal reactions and products. So I hope you can see now that we've looked at cellular respiration and photosynthesis, that they're reverses of each other, that they're the opposites, that the reactants of one are the products of the other, and that they also participate in ATP cycling. One produces ATP, the other utilizes it to create the nutrient that can then be broken down to produce more ATP. So they do go hand in hand.